Hi, everybody. How are we doing? We all nervous for tomorrow? Big day. Big day tomorrow. I'm looking forward to hearing how you've all got on. I hope we all get some really good grades. And I hope whatever grades you get, that it allows you to do biology A level. I think we've got to get usually what, sixes or sevens, so we can do science A levels. Hey, Kira. Hey, CFK. How are we doing? What are you hoping to get? Put it into the chat. What are we aiming for? Morning. Hi. Yeah, it's nerve-wracking. I'm going to be really nervous for you. I'm probably not going to sleep tonight because I'm going to be so nervous for you. But yeah, I'm looking forward to hearing how you all get on. And hopefully we all get some good grades. Paper 2 biology messed me up so bad. Oh, I'm sure it didn't go as badly as you thought. I'm sure it didn't. Sometimes the students that come out and say it was really, really easy haven't done as well as they hoped. And the ones that thought it was hard have done better than they think. Grade 8s and 9s, 8s, 8s for combined, yeah. Hopefully we'll get some 8s and 9s. Okay, right. Yeah, I just want you all to be able to do biology A level. If that's what you want to do, then hopefully you'll get the grade necessary. And then it doesn't matter what you get, right? As long as you can do the A levels that you want to do. Yeah, being nervous shows you care. I do care. I really do care. Yeah, I'm not going to sleep. Okay, right. Should we make a start anyway? Let's take our mind off of it for a little bit. Let's do some year 12 biology. Today's lesson, I'm actually really looking forward to it because DNA is one of my favorite topics. So I thought what we could do is like a little recap of GCSE DNA and then just add a few extra bits on. Because a lot of the A-level stuff on DNA is the same as what you've already learned. As always, there's just a few more details. You only need a 4 to do A-level biology. That's quite low, isn't it, to get onto the course? I mean, you're definitely going to get that right. But you want to go into medicine, yes, you're going to need a higher grade for medicine. Okay. Right, let's annotate this image together. So firstly, where is DNA found in the cell? Now, because we're doing A-level year 12 biology, we can be a little bit more specific. So obviously we say it's found in the nucleus, but can anyone remember which part of the nucleus? Because at GCSE, we just say it's found in the nucleus, but the nucleus, as we now know, has a nuclear envelope, it has a nucleolus, it has nucleoplasm. Can anyone remember exactly which bit the DNA is actually found in? Jazz is talking about chromatin. Yeah, we're going to get onto that in a second. Chromatin. But which bit? Is it the nucleolus or is it the nucleoplasm? It's not the nucleolus. It's not actually in the nucleolus. The nucleolus, let me do a little picture. So if this is the nucleus, the nucleolus is like a small circular region in the nucleus. But the nucleolus, don't forget, makes ribosomes. So that's not actually where we find all of the DNA. Yeah, so Kira and Aisha are saying it's actually in the nucleoplasm. Okay, so that's specifically, let's just link that down. So it's actually in the nucleoplasm. Now, in the nucleoplasm, we've got our chromosomes. Just remind me, how many chromosomes in a human body cell? Yeah, it is the nucleoplasm, Aria. Well done. How many chromosomes in a human body cell? Easy question, hopefully. In a human body cell. Good. There's different ways of saying it. So Zafia says 23 pairs. Zainab, it's not quite 48. You just deleted that then, I saw. Yes, it's 46 or 23 pairs. So let's just annotate this on as well. 23 pairs or 46 chromosomes in a human body cell. Now, a couple of you mentioned before the term chromatin. So chromosomes are made of chromatin. What is chromatin? What is chromatin made from? And we'll just get all this down as a little starter. So what is chromatin made from? Because chromosomes are made from chromatin. Perfect, Angelina. So chromatin is made from DNA wrapped around histone proteins. Well remembered. I think I'd probably talk to you about toilet rolls, because that's how I always think of it, is like the DNA is like the toilet paper, but it needs to be packaged and wrapped. So we wrap it around histone proteins, which are like the toilet rolls. Did I talk to you about toilet rolls, guys? I forget what I've said to which class. What is a histone again? So it is just a protein, but it's a protein that DNA is wrapped around. So it's like a spherical protein or a globular protein that DNA is wrapped around. You love these analogies. Oh, I've got plenty of them. We've just got to be careful, because I use analogies to try and help you understand it, right? But then I'm always worried that students are going to write it in an exam. Like they're going to start to say, oh, histone proteins are like toilet rolls, which isn't going to be great, okay? So it helps you to understand it, but it's not for an exam. Have I had any surgery before? No, I've been quite lucky to be. I've never even broken a bone, Aria. Never had surgery, never broken a bone. I've been very lucky. Okay, so then we've got our DNA. So chromosomes, let's just piece it all together, okay? Chromosomes are in the nucleus. They're in the nucleoplasm. They're made from chromatin, which we know is DNA wrapped around these histone proteins. If we look at DNA is split into genes. So my next question for you is, what is your best definition from GCSE of a gene? What is your best definition of a gene, guys? And you can just use your GCSE one and we'll write it down. Obviously, DNA contains loads of genes. We can only see 2 here on this section of DNA, but in reality, there are thousands of genes. So person 1 says gene is a code for something like hair color. Yeah. Angelina says it's a section of DNA that codes for amino acids that make up a protein. Brilliant. Talia says a short section of DNA. Yeah. So it's a section of DNA, and that could be like one marking point. But we also need to say that codes for, and if we give the full definition like Angelina did, it codes for a sequence of amino acids that make up a protein. That's the best definition. Okay, and sometimes at GCSE, we can just get away with saying it codes for a characteristic, but actually what it's coding for is a protein, okay? Because the DNA determines the amino acids, and then the amino acids make the protein, and then that protein will determine a characteristic like your hair color or your eye color or your skin color or something like that. But we're going to say codes for amino acids that make up a protein, okay? Is the nucleoplasm in the nucleus? Yeah, so you've got the nuclear envelope, which is around the outside. It's filled with nucleoplasm. I suppose it's a bit like the cytoplasm of the cell. So the nucleus is filled with nucleoplasm, but there's also the nucleolus inside the nucleus, which if we go all the way back to lesson 1, we said the nucleolus's job was to make the ribosomes. Okay, right. Let's move on then. So we've kind of got this summary going on here. We can kind of see where the DNA is and how the DNA is organized, but we're going to focus on DNA. So you will have all done different amounts of this. It depends whether you did combined science or triple science. It depends whether you did higher or foundation. I'm guessing most of you did higher. But I just was interested to know, how much detail did you do on the structure of DNA? And we're going to use this slide, a bit like the last one. We're going to use it to make a little summary. So we're going to annotate it. Anything you know about the structure of DNA, can you put it into the chat for me? Yeah, so if you did higher and you did triple, you probably did quite a lot. Kira obviously did quite a lot because you've got some good detail already there, Kira. Yeah. Okay, let's get this down then. So we've got a few good comments in the chat already. So DNA is double-stranded. And linked to that, as Halima said, it forms a double helix. So we describe the structure as a double helix. And you can see this on the diagram. You can see the two orange strands. So you can see it's double stranded. And you can see that those strands are kind of twisted around each other to form this double helix. Okay. Guys, I think you've pretty much got everything there that I wanted you to know. So Angelina says it's made from nucleotides, which we're going to look at in a bit more detail. Okay, so it's made up of nucleotides. And someone as well told me in the chat that nucleotides contain a sugar, a phosphate, and a base. Okay, a sugar, a phosphate, and a base. CFK has named the bases. Well done. At GCSE, did you just learn the letters or did you actually learn the names of the bases? Can you let me know? Because I think at GCSE you can get away with just the letters, right? But did you actually learn the names? Person one says just letters. Yeah, we just learned the letters. A, T, C, and G. Let's try and learn the names then now, because for A level we are going to name them, okay? And you can let me know if you've heard of them before. So the A is for adenine. The T is for thymine. The G is for guanine. And the C is for cytosine. Now, I don't really have any tricks for you to remember these. I feel like you're just going to have to keep saying them and keep writing them to learn the names off by heart. Yeah, adenine, thymine, cytosine, and guanine. Good, Kira. Yeah, so Aria says you've heard the names. Yeah, I think we probably used the names before, but I will have told you that you didn't have to remember them for GCSE. Right, Zion says complementary nitrogenous base pairs. They join together with hydrogen bonds. Right, so do we know the base pair rule? Do we all know which bases pair together? Because you can see in the center, the bases form, it's almost, oh, here's another analogy for you. It's almost like a ladder, right? A twisted ladder. And the bases are like the rungs of the ladder or the steps on the ladder. So A, or adenine, always pairs with T, and G, or guanine, always pairs with C. A, T, G, and C. But there is an exception during protein synthesis where thymine changes to uracil. Yes, so Aria, uracil is a base that we find in RNA. So what you're talking about there, which is correct, is when we're making RNA, we have to use a uracil instead of a thymine because there's no thymine in RNA. It's uracil instead. So well remembered there, Aria. Now we do have multiple ways of remembering the base pair rule. Does anyone want to share one of your little tricks? I'm sure you've got a trick for remembering ATGC. There's loads of different things that I've heard over the years. Or do you just remember it? I don't know. I've heard lots of different ways. For example, I've heard apple trees and garage cars. G and C look similar. Yeah, person one, I've heard that. If you look at the letters, A and T are all straight lines, whereas G and C are curved lines. So I've heard that before, that the straight letters go together and the curly letters go together. CFK says, I just remember it. Yeah. Or what did I used to say? At the golf course. So at the golf course. Or I've heard apple trees and ginger cookies. There's all sorts of different things, okay? But yeah, they pair together, and this is because their shapes fit together, okay? So the shape of the adenine fits with the thymine, and the shape of the guanine fits with the cytosine. Now, who was it? Someone even told me the name of the bond between the base pairs. So let's just get this down and then we'll move on. So there are hydrogen bonds between the bases. Okay. So the A pairs to the T with hydrogen bonds, and the G pairs to the C with hydrogen bonds. Guys, what do you know about the strength of a hydrogen bond from chemistry? Amina towards grey cat. That's a different one. Right, yeah, A says weak. They are weak. They are weak. What do you call them, like weak intermolecular forces? So Jawahir, they are easily broken. They are easily broken. Okay, yeah, they're weak intermolecular forces or weak bonds. We can get away with saying in biology. So they can easily be broken, and we're going to think about why that is important in today's lesson. They're not actually covalent bonds, CFK. They are intermolecular forces. So they are quite weak. Covalent bonds, on the other hand, are quite strong. Okay. We're actually going to talk about covalent bonds in a second, because there is one in DNA that we're going to learn for A level. Okay. So this is pretty much like what we learn at GCSE plus a little bit more, I would say. Like, you probably didn't know the names of the bases. You probably didn't know there were hydrogen bonds between the bases. So we're starting to build it up a little bit. Okay, I'm going to introduce what I think is a new word now. We're going to say DNA is a polynucleotide. Now, if you see the word poly ever in biology or chemistry, what does poly actually mean? A polynucleotide or a polypeptide. What does poly actually mean? Multiple, many, right? So, or lots of. So polynucleotides basically translates to DNA is made from many nucleotides. Polynucleotides, many nucleotides, more than one. I'd say it's more than two though, right? Poly means many. Usually if we're just talking about two, we say di. If we're just talking about three, we say tri. So poly means more than that. Poly means many, okay? So it's a polymer. It's an example of a polymer. Guys, it's been a long time since I did chemistry, but in chemistry GCSE, do you learn the definitions for polymer and monomer? And if you do, can you tell me what you learn in chemistry? So DNA is a polymer. Yeah. So what would you say in chemistry? What would you give for a definition? Or would you even be asked that in an exam? Aria says a polymer is a long chain of monomers, whereas Jawahir says monomer is singular. Yeah. Talia says a polymer is a long chain. Oh, that's a really good one, Talia. Let's write that down. A polymer. A long chain of repeating monomers. Monomers is a small chair. A chain. A small chain. It's actually not a chain. A monomer is not a chain. A monomer is just a single unit. And I'll put in brackets from which polymers can be made. So a monomer is not a chain, just to clear that up. A monomer is just one single unit, and then a polymer is many of these single units linked together to form a chain. Yeah, so Jawahir, that would be a good definition as well. A polymer is a long chain of the same monomers. Yeah, or a long chain of repeating monomers. Okay, so have a look at DNA here. Now, I've given you a more complex diagram. Now, remember we said DNA is a polymer. So it's a chain of repeating monomers, but the monomer is called a nucleotide. which is why we say DNA is a spell that wrong. DNA is a polynucleotide. Okay, because it's made from many nucleotides. We get it. Nice. Okay, so can we label all these bits? Should we look over here? We've got the component parts of a nucleotide here. So the blue circle is the phosphates, whereas the pentagon is called deoxyribose. What is deoxyribose? What type of molecule is that? Because we know there's a phosphate group, we know there's a base, which we can see here, but what part is the deoxyribose? Good, Kira, this is a sugar. Good. It's actually, it's called a pentose sugar. Does anyone know why? Good, Zainab, a monosaccharide. That's some good language there. A monosaccharide. Yeah, it's a simple sugar. So Zainab says 5 sides, not a bad guess. Not a bad guess because it's a pentagon, right? Pentagons have 5 sides. It looks like a pentagon. It's actually not the number of sides. It's because it contains 5 carbons. But you're right, it looks like a pentagon. But the reason we call it a pentose sugar, or we say deoxyribose sugar is a pentose sugar, is because it has 5 carbons, okay? Does anyone know what kind of sugar glucose would be then? I'm extending you a little bit here, but we know the chemical formula for glucose, right? How many carbons does glucose have? Good, Zainab. And is it Tabor? Yeah, glucose has 6 carbons. C6H12O6. So that would be an example of a hexose sugar. You're going to learn a lot about glucose in A-level, I'm telling you now. You're going to have full lessons just on glucose. But that would be a hexose sugar. Now, the sugar in DNA is not glucose, okay? It's deoxyribose sugar, and it's only got 5 carbons, so it's a pentose sugar. Okay, so let's just draw together a single nucleotide. Here we go. So this is one nucleotide, a single nucleotide. So we've got the phosphate, we've got the deoxyribose sugar, which is our little pentagon shape, and then we've got our base. Yeah, exactly, Aria. DNA stands for deoxyribonucleic acid. Okay, so that's our nucleotide. But what we need to look at on this slide, oh yeah, it's on the next one, isn't it? What we need to look at on this slide is look at the overall structure, okay? It's double-stranded. So you can see really nicely here that there are 2 rows of nucleotides side by side, okay? Let's just put a box round a couple. So we've got a nucleotide here, and we've got another nucleotide here, okay? So it's double-stranded. And remember, it twists to form this double helix. The 2023 paper, you had to circle nucleotides. Yeah, I've got a question actually in this lesson. Sometimes they just ask you to circle a nucleotide or put a box around a nucleotide, and they're just checking that you know what 3 parts are in the nucleotide. Okay, so just to quickly check what you've remembered, can you put into the chat, what is A, what is B, and what is C? Yeah, they might have asked you to do that at GCSE as well. If you did triple science, they might have asked you to circle a nucleotide. So what is A, what is B, and what is C? Tabor, can you name the pentose sugar? Can you specifically name the 5 carbon sugar? Because there are different 5 carbon sugars, so it's better if we can actually give the name. Good. So it is a pentose sugar. It's a 5-carbon sugar. But if I was you, I would always name it in an exam, because RNA, for example, RNA is also containing a pentose sugar, but that's ribose sugar, not deoxyribose sugar. So if we're specifically labeling a DNA nucleotide, I would name the specific sugar. Okay. Right, so this is our monomer, which remember is our single unit, okay? And we need to remember it's called the nucleotide. What we're going to do now, and I'm hoping you've got some space in your workbook, we're going to try and draw 2 nucleotides together so we can talk about the bond that actually holds them together as well. In fact, we might even try and extend it and draw 4. But for now, I want you to try and copy. It's quite tricky. It takes a bit of practice to draw your own nucleotide. Aria, am I getting ahead of myself? No, I want to do it here. Just checking I hadn't got it on the next slide. I want you to try and draw 2 nucleotides like this. Okay? I want you to try and draw 2 nucleotides like this. So we've obviously got the phosphate, the sugar, the base. The phosphate, the sugar, the base. But does anyone know the name of this bond? Now, I wouldn't expect you to, but this is the bond that joins one nucleotide to the next. It's not joining the bases, so I'm not asking you for hydrogen. It's joining the deoxyribose sugar of one nucleotide to the phosphate group of the next. And this is a strong bond. Tabor, well done. This is called a phosphodiester bond. Phosphodiester bond, right. We've not learnt this before, I don't think, unless you went really ahead at GCSE. But phosphodiester bonds are strong covalent bonds. And they join one nucleotide to the next. Now, you'd be surprised how many students, even in year 13, get these bonds mixed up. They forget what that bond is called, or they tell the examiners that it joins the bases together. It is nothing to do with the bases. The bases are held together by hydrogen bonds, which are weak, okay? But the sugars and the phosphate groups are held together by phosphodiester bonds, which are really strong, okay? So these bonds are giving DNA its strength. They're making it really stable. The backbone, have I used that term yet today? Do we know that we can refer to this as the sugar phosphate backbone? Okay. The backbone is really strong because the backbone is held together with phosphodiester bonds. There's too many bonds. Honestly, Zainab, like every lesson you're going to learn a new bond. Biology, like all we talk about is bonds. After 2 weeks of year 12 biology, you're going to have met so many different bonds and you do start to mix them up, okay? But in DNA, we've got hydrogen bonds between the bases and we've got phosphodiester bonds in the sugar phosphate backbone. Okay, the hydrogen bonds are weak. So we're going to talk about them, being easily broken, but phosphodiester bonds are strong. So they give DNA a lot of strength and stability, okay? And remember, they're going from the sugar, the deoxyribose sugar, to the phosphate group. Do you need to know the exact name? Yes. So A says, which part is known as the sugar phosphate backbone? Let me go back and see. So you see here, it's basically the sugars and the phosphates, right? So this is one sugar phosphate backbone, but this is another sugar phosphate backbone because it's double stranded. So there are 2 sugar phosphate backbones. And as the name suggests, it's just the bits with the sugars and the phosphates in, okay? And you can see, should we highlight some phosphodiester bonds? Look at my pen, guys. I'm hoping you can see this. These things that I'm shading red, these are all phosphodiester bonds. Okay. So they're joining the sugar phosphate backbones together. Shouldn't the second one be upside down? It should, shouldn't it? It's not the best diagram. I mean, I could try and draw how it should look, but I think I might struggle drawing two strands. Zainab's just noted something. So it should... Oh, this is where I struggle drawing DNA upside down. This is testing me now. Yeah. Can you see? This second strand here, the sugar is upside down. Can you see? The pentagon is the opposite way around. It should be upside down. Yeah, they are. The two strands are anti-parallel, we say. Anti-parallel. Anti-parallel. So yeah, if you look at them, it goes phosphate, sugar, but the other one is going the other way around, sugar, phosphate. And if you look at my pentagons, the second strand, the pentagon sugar, is flipped upside down. Yeah, this one is kind of correct. Like, it's kind of correct, but the sugar is the wrong way up, which I think is what Zainab noted. If you look at the second strand, the phosphates are correct, but the sugars are the same way up in that diagram, and that second strand should have the sugars upside down. I'm going to have to get that picture redrawn, I think, so it doesn't cause confusion. Okay, right. So let's do some more on the base pair rule. Firstly, super quick, can you tell me the sequence of bases that you would expect to find on this second strand of DNA? So the yellow is a simplified sugar phosphate backbone. Very simplified, obviously, because we've just shown it as a yellow rectangle. The bases are in the center. They form the rungs of the ladder. So quickly give me your complementary bases. Well done. Just checking. Nice. Nice and easy. Yeah. So we can see our two sugar phosphate backbones. Bases are in the middle. I'm going to do this. And you're going to tell me what I'm doing. What am I telling you here, guys? What bonds am I drawing here? And what am I telling you about the numbers of bonds? Good. Anime, good. What do you notice about the numbers? So we know they're hydrogen bonds. They hold bases together. Kira's noticed, yeah, the GC has 3 hydrogen bonds and the AT only has 2. Okay, let's write this down because this is something else we need to know for A level. So we've got 2 hydrogen bonds between adenine and thymine. We have 3 hydrogen bonds between guanine and cytosine. Yes, and we do need to know this level of detail, okay? So we just keep adding more on. So we've got phosphodiester bonds in the sugar phosphate backbone. We've got hydrogen bonds between the bases, but we've got 2 hydrogen bonds between adenines and thymines, and we've got 3 hydrogen bonds between our guanines and our cytosines. Have we covered everything yet? How do you feel? I think we've learned a lot more than GCSE. Guys, you don't have to be able to draw the bases. So don't panic. When you see these diagrams on the right-hand side, these are the actual structural diagrams of the bases. You don't have to draw them, okay? So don't panic. That's just to show you that they have different shapes. They have different structures. Is there a certain amount of adenine? Would there be the exact same amount? Yes, exactly. CFK, we're going to talk about that later. Because adenine always pairs with thymine. So wherever there is an adenine, there would be a thymine. So there would be the same number. Can you explain the differences between the types of bases? Yes. So if you look at adenine and guanine, these have got what we call like a double ring structure. And these types of bases are actually called purines. Now, this has been taken off the A-level spec. So you actually don't have to know the structures of the bases. You don't have to know the differences between the bases, okay? It was on the old A-level spec, but it's not on the new A-level spec. But the thymine and the cytosine are single ring bases. So they're shorter, right? Now, these types of bases were called pyrimidines. But luckily for you, you don't have to know which are purines, which are pyrimidines, which are double ring bases, which are single ring bases. It was taken off the spec in 2016. Okay. So when I say old spec, I'm talking like old spec. It used to be on before they changed the spec. The last time they changed the spec was 2016. So we've been on the same specification for quite a while now, which means it might change again soon. But yeah, basically they just have different structures. So a double ring base always pairs with a single ring base. So adenine always pairs with thymine, and guanine always pairs with cytosine. So can you see it's a double ring always pairs with a single ring? And that's making the rungs of the ladder will all be the same width, because you always have 3 rings together, if that makes sense. Double always pairs with single, so they're always 3 rings in length. They better not change it for us. They're definitely not going to change it for you because it's too late now. They have to give like 2 years notice because obviously teachers need time to read the new specs and plan lessons and stuff. So if they were to announce that they were going to change it like this year, those changes wouldn't come into effect until like 2027 probably. OCR changed the spec last year, only very slightly though. They didn't do a full new spec, Zainab. They didn't do a full new spec. They just made some very minor edits to OCR. And most of the things, they took out a few bits and bobs. So it's not too bad. Okay. Right. This is for you now. Guys, you might think, oh, this is repetitive, but you've got to learn it. One of the things about biology that makes it harder than some other subjects is the amount of content and the amount of things you've got to remember. Now, this could be a 6-mark question, and it would be a question at the back of paper 1 where you get some full written response questions worth 5 or 6 marks. And they can just say to you, even without the diagram, right? They're probably not going to give you a diagram. They can just say to you, describe the structure of DNA for 6 marks. Sometimes they say describe the structure and function of DNA for 6 marks. So I want to try and get more than 6 marks here. I think we can get like 10 bullet points into the chat. But I want you to all try and include something new that you've learned today, not just GCSE level. Do they mark the 5-6 marks the same as GCSEs? No, they don't, Kira. So at GCSE, it's a little bit easier because they put you into a level, and it can be quite easy to get full marks at GCSE. Whereas at A level, it's like specific bullet points that you have to say. So it is more specific at A level. Okay, let's start writing down. Can you keep going in the chat, guys, and we'll see how many we can get. Good. Good. You're giving me the A-level stuff, which I like. What did we say it was called, Guy? Like the whole, what's the name of the polymer? I want that as well. Good, Kira, because that can get you a mark, is a polynucleotide. That would probably be the second thing. I'm just trying to go in a logical order. So I'd say it's got a double helix structure, it's a polynucleotide, or it's made from many nucleotides. The nucleotides contain, and we've got to be specific, phosphate, deoxyribose sugar, and a base. Now, someone said earlier in the chat, you can refer to them as nitrogenous bases because the bases contain nitrogen. So if you ever see that nitrogenous base, it just means the base contains nitrogen. And I have actually seen an A-level question where they've asked students to name the part of DNA that contains nitrogen, and you had to say the base. Okay. Okay, so there are complementary base pairs. And to get this mark, you'd probably have to give the base pair rule. And I'm always going to write the name as well, rather than just the letter, because sometimes the letters aren't enough. For example, if it says name the bases, if you give the letter, you will not get the mark. So it's better to just name them. Yeah, bases contain nitrogen, right. So A-level stuff. Bases are held together with hydrogen bonds. We could even say 3 hydrogen bonds between. Now, because I've already named the bases, now I'm going to abbreviate them because I've already named them once. So that's okay. If you abbreviate something, name it once, and then you can abbreviate it. Phosphodiester bonds between adjacent nucleotides. I love that. Love how you've described it. There are phosphodiester bonds between, not between the bases, because that's hydrogen bonds, but between adjacent nucleotides. That's a really good way of saying it, because adjacent means next to each other. So there are phosphodiester bonds between the nucleotides that are next to each other. Have I missed anything? 2 sugar phosphate backbones. Good. Yeah, you could say 2 sugar phosphate backbones, and they are held together with phosphodiester bonds. Did anyone remember what I said about the strands? Ran out of space. Yeah, sorry. I mean, this would be a 6 marker, so we wouldn't have to say all of this, right? But you'd need at least 6. Good. Chissy's remembered. The strands are anti-parallel. I didn't know whether anyone had picked up on that term that I use. The strands are anti-parallel, which basically means they run in opposite directions. Anti-parallel. Well done. Can't see it on this diagram. Again, on this diagram, the second backbone, the sugars are the wrong way up. So the sugars should really be this way to make it accurate. Okay, so try and remember that for me, that they are anti-parallel. For the 6 markers, can you write in bullet points? Oh, for sure. My whole exam paper would be written in bullet points, apart from the essay. Remember, if you do AQA, there is an essay, a 25-mark essay at the end of paper 3. That's the only question that I wouldn't answer in bullet points. Every single other question I would use bullet points, because to me, as I'm writing my answer, I'm thinking about the mark scheme and I'm thinking about the marking points, and just bullet pointing them out helps me to see like how many marks I'm getting. Your teacher said you're not allowed to do bullet points. Why not? Examiners love bullet points. It makes it easier to mark the students' work and actually see whether they've hit the marking points. Do I do a lesson? Yeah, in the main course, we'll do at least, well, we'll do more, but I've already planned a full hour and a half on essay writing, and then we do more essay writing in the Easter revision course and the cram course as well. We do essay writing. I don't know why your teachers are telling you you can't use bullet points. That makes no sense to me. Can you still use bullet points for 2 or 3? You can use bullet points for 6 markers. I mean, are your teachers telling you not to use bullet points because your bullet points are too brief? Like, if you wrote double helix, I mean, I wouldn't do that. I wouldn't just write base pairs. I wouldn't just write anti-parallel. Do you know what I mean? That would be too vague. So maybe your teacher's telling you that because they're worried that your bullet points would be too brief. But if each of your bullet points is a key piece of information, like the strands are anti-parallel and there are complementary base pairs, there's no problem with that. That's so crazy that your teachers are telling you that. What did I study at uni? I did biology at uni. My degree was just biology. The reason I did biology was because you could pick from all the modules if you did biology. So you could pick exactly like the modules you were most interested in. And then for my A-levels, I did biology, chemistry, psychology, and sociology. But biology was obviously my favourite. Okay. Right, let's do a couple of questions then just to finish, guys. Your physics teacher gave you a detention for using a formula triangle. Yeah, physics and math teachers do not like formula triangles. I'm sure you've learnt this, but I use them in biology. You know, I'm not a maths teacher. I didn't do maths A level. So if I need to use one for like working out magnification or rearranging like speed equals distance over time, I'm not ashamed. It helps me. As long as you get the marks, you can do whatever helps you. That's bad. Why are they so? I suppose they think that if you're using a formula triangle, you don't understand how to rearrange an equation, which is more important in maths and physics that you understand it, right? CFK is way ahead of the game. Right, I'm going to throw some A-level questions at you. So these are kind of maths questions, but they're testing you on your knowledge of DNA and the base pair rule. So the first one we're going to do together, I'm going to show you how you do it. And then the second one, you're going to do it. Do you get an equation sheet in A-level biology? No, because there's not enough equations that you have to know. But sometimes they give you equations in the exam, like if they want you to work out standard deviation or the index of diversity, they give you the equation in the question. But you don't need a whole formula sheet because there's not enough equations. Right. Suppose the percentage of cytosine in a molecule of DNA was 34%. Calculate the percentage of thymine. So cytosine always pairs with guanine and adenine always pairs with thymine. Now they've told us that cytosine accounts for 34% of the bases. So what I know is that guanine must also account for 34% of the bases because cytosine pairs with guanine. So wherever there is a cytosine, there is a guanine. So there's going to be the same percentage of cytosine and guanine, okay? So now I know that C and G together accounts for 68% of the bases, okay? Now, obviously, 100% take away 68% leaves 32% of the bases unaccounted for. So now I know that 32% must account for adenine and thymine. Yeah? And adenine and thymine, there must be the same percentage of both because they pair together. So the percentage of adenine must equal the percentage of thymine. So all I need to do is divide 32 by 2 to get 16%. So there must be 16% thymine, which is my final answer. Okay. Who understood what I did there? What I'm going to suggest is, so I can see who's understood it and who hasn't, have a go at question 2 using the same, I mean, you can do it faster once you get it, right? But while we're still learning, try and set it out in a similar way to me. See who can get it right. And then if we don't get it, I can go through it again. This time we've been told the percentage of adenine is 18 and we want the percentage of guanine. Now remember, adenine always pairs with thymine and guanine always pairs with cytosine. Yeah, there's simpler ways of doing it. I'm just trying to explain it, seeing as this is the first time we're doing it. Guys, I think you're getting the answer right. So adenine accounts for 18%. We're told that in the question. So we know that thymine must account for 18%. Okay, so together, if we add them up, adenine and thymine accounts for 36% of the bases in the DNA. So that leaves us with 64%. So 64% must account for guanine and cytosine, but they are going to be present in equal quantities. So I just need to divide that by 2 and I get 32% guanine. Yeah? Okay. Easy? They like these questions because it's maths, and 10% of the biology A-level paper has to be maths, right? That's the rule. The examiners, when they write the papers, they have to make sure that 10% of the marks are for maths. Now, the maths isn't that hard. It's just like GCSE level maths. But what makes it hard is it's maths in a biology context. So here they're trying to get you to do some really simple maths. It's just like adding and dividing, right? Really simple, but you've got to understand the biology to do the maths. And that's why maths in biology can get a bit tricky. But if we know the base pair rule and we understand the structure of DNA, the maths here is easy. How far before the exams do they write the papers? 2 years. So let me think about it. Yours will already be written. It will just be getting like checked at this point. If you don't get a 6 in maths, can you still do A-level bio? It depends on your school or college. Like some schools say 5 for maths, some say 6, some might even say 7, I don't know, but it depends. Okay, last question. Yeah, I'd say grade 6 would be, I can understand why they're saying grade 6 is recommended. The maths isn't hard. Like, you're not going to be doing crazy maths like you do in A-level maths, but quite often it's like figuring out from the question what maths you actually have to do, because there's quite a lot of writing in the maths questions as well, and tables and graphs and things. There's a lot of unit conversions. Standard deviation, yes, we have to do statistics. So we do standard deviation, we do chi-squared, we do Spearman's rank, like statistical tests to check the significance of the results. There's a whole module on statistics. Right, draw a box around 1 nucleotide. Let's do this super fast. You could do any. I'm going to do this one. You could do one on this side. It doesn't matter, as long as you've got in your box a phosphate, a sugar, and a base. Okay, a phosphate, a sugar, and a base. Okay. So that time when the GCSE truck went missing, did they have a spare or use the one for the next year? I don't even know about that story, Zainab. I haven't actually heard of that, so I don't know what happened there. Right, use the letters in the diagram. Can you put your answers into the chat for this, please? I'm just going to rub off some of my boxes so you can actually see all the letters. So We've got A, B, and C labelled on the diagram, and you need to use those letters to tell me which part is a sugar and which part contains nitrogen. Good. So B is the sugar because it's deoxyribose sugar, right? But we only need the letter. It said use the letters, so put B. This is where reading the questions is so important. Like some students might have written deoxyribose sugar. They're not going to get the mark because the question was actually asking for the letter on the diagram. Contains nitrogen is A because we can describe that as a nitrogenous base. It's a base which contains nitrogen. The sequence of bases on one strand of DNA is important for making proteins. What is its role? So the sequence of bases in DNA determines what? We said it right at the start of the lesson when we did our definition of a gene. Good, Jazz. Good, Angelina. The sequence of bases in DNA is important in protein synthesis because it codes for the sequence of amino acids in the protein. Good. So the TTC would code for one amino acid, and then the next three bases would code for another amino acid. Remember, it's 3 bases. Do you remember that from GCSE? 3 bases codes for 1 amino acid. Right, how are the 2 strands of DNA held together? These questions are nice. And by the way, these are exam questions. Yeah, I haven't just like made them up. These are all based on real past exam questions. So some of them are nice. Okay, be careful, Zainab. This is asking how are the 2 strands held together. So we're looking for the bonds between the bases that hold the strands together, not one nucleotide to the next. So this would be hydrogen. Does that make sense? Because the phosphodiester bonds join one nucleotide to the next, but the 2 strands are held by the bases and the hydrogen bonds between the bases. Give one advantage of DNA molecules having 2 strands. What does it allow for? Think back to GCSE. Think back to the cell cycle. Why do we need 2 strands of DNA? Anyone got any ideas? Not more protein because only one strand actually acts as a template. Only one strand is needed for protein synthesis. Okay, so Jazz is on the right lines. Jazz is saying mitosis. Lily is saying replication. Yeah, it allows the DNA to be replicated. I'm going to put before cell division. Because mitosis and meiosis, this is bringing back bad memories now of GCSE. Mitosis and meiosis, before a cell can divide, all the DNA has to be replicated. And the way that works is the 2 strands separate, and then we use those 2 strands as templates to build the new strands. So having these 2 strands is basically what allows for DNA replication. Okay, and we'll do at A level, you do a whole lesson or maybe even 2 lessons just on the process of DNA replication. So you'll see it then. Right, if you want to stay on, it's up to you, you don't have to, but I've got some information. I know some of my other students were asking for this information. So if you do want to join our full course, I'll go through it as quickly as I can. And then if you've got any questions at the end, you can ask me. If you do want to join our full course, I suppose this is going to depend like results day tomorrow. If you get on to A-level biology and you want to make the best start possible, starting in September, we'll obviously be launching a year 12 biology course. So specifically for students starting year 12. It goes all the way from September to June. So you get 80 hours of lessons because you have 2 lessons per week. with me, okay? So 2 hours of lessons per week with me. Each lesson has a homework, which are exam style questions, and you get a video solution with the homework, which is basically me going through the homework and explaining the mark scheme and all of the answers. And at the end of each month, you'll also get an assessment, okay? Yeah, so that's how it works. I'll just go through it all and then I'll try and answer all your questions. This is not our prices, okay? This is us saying to you, a lot of people choose to get a private tutor. To be honest, a lot of people leave it until it's too late. They leave it till the end of year 13, they panic, and then they get a really expensive private tutor. Now, obviously, if you've had private tutors before, they can charge you like 40, 50, 60 pounds per hour, and it can get really expensive. Now, we've done the maths. If you got a private tutor for the full year and you did 80 hours, it could end up costing you nearly £5,000 for the year, which is just ridiculous amounts of money that we don't want any of you to have to pay, okay? There are other problems with getting a private tutor, like you might not know them, you might not know like how good they are, it's a little bit of a risk, you might not get homework, you might not get workbooks, you might not be able to fit it in, you might have to travel to their house, etc. They might have to travel to your house. It can be quite difficult to get a good private tutor that kind of fits in with everything else you've got to do, all your other commitments. So this is why we created My Headspace. Obviously, you all know about us now because you're on the summer school course. But our aim is to offer you the cheapest tuition and support. In fact, I don't even want to call it tuition because we teach the full course, right? It's not just extra tuition. If you join us from September and you go all the way through to June, we will cover absolutely everything for year 12, including the practicals, exam technique, maths, everything. So this is kind of what you could possibly pay with a private tutor. But if you come with us, this is the full price for the full year. 80 hours of lessons, 80 workbooks, 80 homeworks, 80 video solutions, all of your monthly assessments. But because you've all been to the summer school, we can give you a much better price than that. So you can get a 30% discount. So let me just explain what that means. You're looking at the orange box, okay? If you take biology for the full year with me from September to June, it's £397 for all 80 hours for the full year, okay? Or 597, I think that's if you also get a private mentor. We offer a mentor service, which you might know if you've been on our GCSE courses. So you can get a one-to-one personal mentor as well. So yeah, that's with the discount. So it's 33% off because you've been in the summer school, right? If you want to unlock this offer, get the discounted price, make sure you are set for September, not leaving it till the last minute. There's a link in the description of the YouTube video. And if you click that link, I think it says something like summer discount or course discount or something like that. If you click that link, it will take you straight through to our WhatsApp and you just send the code word summer 2024. Even if you just want some more information, you've got more questions, you just want to know a little bit about it. Yeah, just go onto the link in the description. I think I'm telling you the right thing, just under this video. Oh, and like the video as well while you're there, guys. That would help me out. Like the video, click on the link. It takes you to WhatsApp, send the code word. They'll know then that you've been in my lessons, and that's why you can get 33% off. There's a full 14-day money back guarantee. So if you start in September, you do 2 weeks. For any reason you don't like it, you can get your money back within 14 days. So it's no risk. There's other subjects, just so you guys know. There's maths, there's biology, there's physics, there's chemistry. So it might be that you actually just want chemistry, you just want biology. You might want all the subjects. So you can get something called the Ultimate Pass. I don't know whether anyone's heard of this. The ultimate pass is where you get access to all of the subjects. So if you do maths, biology, and chemistry, or physics, maths, and biology, we've obviously got all three sciences and maths for year 12. And this will take the price down even further, because if you do all three of your A-level subjects with us, you can get all three for £977. All three for the full year, if that makes sense. But feel free to just click the link, send the code, and then ask any questions, whether you just want to do one subject or whether you want to do like 3 A-level subjects if you're doing all sciences and maths. Because if you opt for all three, basically the price goes down even further. So you're ending up paying like £2 per hour of lesson. You can pay in installments. You don't have to pay it all in one go. So yeah, you could just pay one installment today. This would be like your first installment today if you went for the ultimate pass and you did biology, chemistry, and maths. That would be all you would have to pay today. Or if you want your own personal mentor, which I can see you're talking about in the chat, mentors just can help you with homework. They can help you with revision. They can give you extra questions. They're just like there for your support whenever you need them. But yeah, it is a limited offer. So there's only so many places that we're selling at the discounted price. So that's what you need to do. I'll leave that. That's the year 12 timetable, by the way. What about 4? I mean, you could do all 4, but are you doing 4 A-levels? Are you going to be doing chemistry, physics, maths, and biology? Will there be this offer next year for year 13? Yeah, obviously I'm showing you the year 12 course because you're going into year 12, but next year we'll have a similar offer, I would think, for going into year 13. Yeah, if you're doing all 4, what I would do, Naima, is just contact them, click on the link, send the summer 2024, and just say, like, I want all 4 subjects. And you'll probably get a really, really good deal. You can get the ultimate pass and it'll work out as like £2 per lesson, probably, for all subjects. Is this just for AQA? Lily, you're going to have to send the code word and ask, because for chemistry, there's like AQA and OCR. For maths, I think it's mainly Edexcel, but the exam boards are pretty much the same. So I think it's kind of applicable to all exam boards, the maths one. Physics, I think it's more AQA focused. Biology is more AQA focused. But biology AQA is very similar to OCR in year 12. It's not in year 13. They are quite different. So we have separate courses in year 13 for OCR and AQA. But year 12 biology is AQA focused. I hope that's answered the question. For biology in year 12, like AQA and OCR are about 90% the same. But in year 13, they're quite different. Okay. I feel like I've just been talking at you for ages. Are there any other questions? What I would do, seriously, guys, because there's a whole sales team, there's a whole customer service team that know all about these courses as well. They know all about the prices, the best discounts. I would just send the code word, even if you're just wanting some more information, just get into a chat. All it will do is take you into a chat with someone and you'll be able to tell them like what subjects you're hoping to do. Maybe contact them tomorrow after your results. What subjects you're hoping to do, what is the best price, the exam boards, all that kind of stuff. Is bio from 6:50 to 7:50? Yes, it is. Yeah, Monday and Wednesday it looks like for bio. 6:50. But the lessons are all recorded as well. So I forgot to mention that. So say you think like, oh, I can't attend the live lesson on a Monday because I've got netball or whatever. The lessons are all recorded, so you can always watch them back at a time to suit you. Obviously, we like you to come to the live lessons because you can use the chat and you can ask questions. But if you really can't, they are all recorded and you have access to those recordings for the whole year. So when you come to revise at the end of year 12, you can go back to the lessons you found the hardest as well and watch them as many times as you like. Okay, right, I'm gonna go, guys. Please get in touch tomorrow. Let me know how you've got on. Obviously, you can message me through social media. Let me know how you got on. If you're pleased with your results, if you're definitely going to do A-level biology, let me know. And I will see you next Monday. Good luck. I'm thinking about you all. You know I'm not going to sleep tonight because I'm going to be worrying about you. So I'll keep my fingers crossed. Good luck. Good luck, everybody. Yeah, exactly, Zafia. Let's keep our fingers crossed for some amazing news tomorrow. Right, bye, guys. Bye.